how we discovered these cytoskeletal proteins for the first time. So first I'm going to ask you, do you know what GFP stands for?
Green fluorescent protein.
And where did you find this first time the green fluorescent protein from?
Was it jellyfish?
It's a gene of a jellyfish where it gives us the green fluorescence. The protein is shining, right? It can fluoresce. So what are you looking at in these pictures is what I would ask. What do you think is being represented?
Internal structures?
Internal structures of proteins that have the labeling of the green fluorescent protein. So you can put these cells under your bacterial culture, under the fluorescent microscope, and you'd be able to locate where those proteins are, the fluorescent proteins, okay? So these are attached. The GFP has been, this GFP fluorescent protein is now attached to the FTSZ product. So FTSZ GFP fusion protein is being viewed by the fluorescence microscopy in a chain of Bacillus subtilis. So where is it located? Where do you think this green protein, which is a link to the FTSZ, where is, you can visualize it in a living cell, in the cell that has been stained with the fluorescence, the fusion protein is being looked at. And where do you think it is lying? Where is it within the rod-shaped bacterium?
In the septum?
In the center, in the center. So you understand that the cells, when they have to do a binary fission, meaning they have to find the center of the cell so that they can lay the cell membrane and the cell wall at that point so that two equal sized progeny cells are produced. So you understand there is a lot of measurement going on in the cell to be able to do a binary fission. A binary fission where two equal sized cells have to be produced. The cell has to determine the center of the cell, and there it would be able to lay down the cell wall, the partitioning there. So FTSZ is the protein that determines the center of the cell when the cells are dividing. This is what this was observed by using this fusion protein and using your microscopy in Bacillus subtilis. Again, in the Bacillus subtilis, in this second picture, you're looking at a protein which is MBL. Remember, we talked about the MBL, and to which you also have GFP. This is a fusion protein. And what do you see this fusion protein looking like? What is your take on it? Do you see a magnified view here of MBL here? What does it look like?
Looks like spirals.
It looks like a helix that is under the cell, inside the cell, running as a helix. And that determines the shape of the bacteria, okay? So having a MBL protein, which would be a cytoskeletal protein now for us, and this cytoskeletal protein actually is a helix that runs along the membrane and that determines that the bacterium is rod-shaped. So what do you think if this MBL was mutated and there was not this MBL around? What do you think would happen to the rod-shaped bacterium?
It wouldn't hold its structure.
So it would collapse into a spherical structure then. You realize that? So bacteria do have some skeletal structures now that we've discovered. And the major way that we have discovered them is because of the fusion proteins that we were able to see with fluorescence microscopy. But there have been other ways to do it, but this was the initial way, and it was really beautifully startling to see this, and you will see that in your research papers also. Anyway, here is another one where you can see a cytoskeletal protein that we call CRES, and that is the red colored or the pink colored protein that is laid only on the curvature of your rod-shaped bacterium. And this is found in the Caulobacterium crescentus. And here, of course, the DNA is stained blue. That's why it's all blue. And your DAP1 is the one that is showing the curvature. The curvature protein is CRES now, the red one. We move on. Oh, sorry. Where am I going? Okay, so so we do have some cytoskeletal proteins now that we can describe, and we can see them their location in the bacteria also. But the question still arose that are there any intracytoplasmic membranes besides these, the membrane that is running as a cytoplasmic membrane? Is there any intracytoplasmic membrane? And yes, we found them now in a lot of bacteria, specific bacteria. Some of these intracytoplasmic membranes, they are continuous with the plasma membrane as if they were infoldings of them. But there are others that are not connected with the cytoplasmic membrane, and they're just found spread out in the cytoplasm. We have, even in some, we have a bilayered intercytoplasmic membrane, which is not continuous with the cytoplasmic membrane. This has been observed in photosynthetic bacteria, and this would almost be analogous to your thylakoids of the chloroplasts that are present in your eukaryotic cells, which also have reaction centers for the ATP formation. We'll talk a lot more about reaction centers and thylakoids and the chloroplast later, but these intracytoplasmic membrane in photosynthetic bacteria would be analogous to these structures that are found in the eukaryotic cells only. But we've also found continuous cytoplasmic membranes, those bacteria that have high respiratory activity. So they need a lot of membranous structures. If they have high respiration rate, and respiration in bacteria is located in the membrane only. So having a continuous intracytoplasmic membrane has been seen, so that longer or larger spaces of membrane will help more respiration rate. And also anaxosomes, and anaxosomes are found in the planktomycetes. These are organelles. These are the site of anaerobic ammonia oxidation, and we will be talking about anaerobic ammonia oxidation as we go along. So we do have some intracytoplasmic membrane representation in these two figures. This is the Nitrocystis oceanus, which has parallel membranes. This is an electron micrograph you can see, and you can almost see these thin lines that is supposed to be membranes that are traversing the whole cell here. And here is another one of those that has a collection of these intracytoplasmic membranes, extensive, lots of them. And this is, of course, the one which has a high respiratory rate. These are almost always connected to the plasma membrane, and they come up as batches of these within the cytoplasm. These membranes are there. So this gives the cell the ability to have a high respiratory rate. Okay. We talk about inclusions in the bacteria. Of course, inclusions are either granules of organic or inorganic material, which the cell is saving for later use when it is going to be starved for those compounds. So storage is done in inclusions. And these inclusions, some of them have a single layered membrane around them. So they are not truly double layered inclusions as they would be in your eukaryotic cells. Here, some of these inclusions may have a single layer membrane. And here the membrane composition of these single layered inclusions, membranes have different compositions. And some of these membrane that enclose your inclusions may be made up of proteins, and some may also be containing lipids in them. And these, as you kind of find a membrane around, even a single layered membrane around the inclusions, these are sometimes now referred to as micro compartments. So these are not the true compartmentalization that you see in eukaryotes, but these micro compartmentalization occurs because of single layered membranes, which are mainly, in this case, when you will see the picture, these are mostly made up of proteins rather than your lipid. So here are representation of some storage inclusions that you may find. This is Bacillus megatherium, which is very well known for its PHB. This is the polyhydroxybutyrate inclusion that it has. So if you look at the figure here, there is a large inclusion here, polyhydroxybutyrate. This is, of course, storage for carbon. The bacteria is trying to store carbon for itself for later use in this inclusion. There will be some that have glycogen storage, some that will have the phosphate storage. Usually it is a polyphosphate that is stored, and you come up with the name of volutin. You may have seen volutin or metachromatic granules, as we call them, when we were staying Corynebacterium in the lab. We used to be able to see them, the phosphate granules. So phosphate granules are also, because phosphate is not found in every environment. The cell has to store it. Some cells are some bacteria have a very good capacity to store them in these volutins, which is the phosphate granules. Then there are some that will store amino acids, and we have the cyanophycin granules found in some. Here in this case, in this chromatium, you are finding these blobs inside the cell. These are all sulfur globules. So these bacteria are definitely sulfur metabolizing cells. Therefore, they have to store sulfur for themselves. And here's what I called the microcompartments, where not a true membrane, but a single layer of protein kind of covering them to make them into microcompartments. And this has been seen as carboxysomes, which is found in your carbon dioxide fixing bacteria. We'll be working on that very soon, as these very special bacteria, halothiobacillus or so, they have carboxysomes where their storage of carbon, they are storing the enzyme, the ribulose bisphosphate, which is the ribisco. This is the enzyme used for carbon dioxide fixation. So these compartments actually allow them to store this special enzyme, which will allow them to fix carbon dioxide. So you will find these microcompartments in those bacteria. Here is the one that other inclusions where in your aquatic bacteria and in your photosynthetic bacteria, in a lot of archaea, you have gas vacuoles. And the gas vacuoles, of course, provide buoyancy to the microbes so they can float up into the air, into the water. And they sometimes aggregate together, and they will be, instead of calling the gas vacuoles, we call them gas vesicles. So these are two different types of gas inclusions in this bacterium called Anabina, which you can see wherever the gas vacuoles or vesicles is. When you do a freeze fracture, they will fracture mainly in the gas vacuole area, and you can visualize them under the electron microscope at that time. Here we have also the magnetosomes. As I told you, that there are some magnetotactic bacteria, and here is a very good representation. This bacterium, Magnetospirillum bacterium, this is a magnetotactic bacterium. They assemble their magnetosomes along the curvature of one side, inside of the plasma membrane. And this, of course, the cytoskeletal protein that we call MAMK helps to arrange these magnetosomes in a long array along one of the sides of the rod-shaped bacterium. Very interesting aquatic bacterium that orients itself to Earth's magnetic field. We move on to what is called the ribosomes, and these, of course, you're very familiar with. You've studied ribosomes in your eukaryotic cells also. So it is a complex structure composed of RNA and protein. So RNA is a structural part of your ribosomes, and this is, of course, this unit is needed for protein synthesis. It is composed, the entire ribosome, if you look at it, in bacteria and archaea, the ribosomes are categorized as 70S, whilst in the eukaryotic cells, they are considered to be 80S. And S stands for Swedberg unit. What does it represent when you want to describe it as to what the Swedberg unit measures? What would you say? Any ideas?
Is it a unit of weight?
No, it's not a unit of weight. It is a sedimentation coefficient of the unit. That means when you spin these in a high-speed rotation, they come at a certain level. So that is their sedimentation coefficient. So the sedimentation coefficients are different based on not only their weight, but their structure also. Okay. So definitely we have differences in your ribosomes of eukaryotic and your prokaryotic. But within your eukaryotic cells, you will find the 70S ribosomes, where would you find them? Do you find 70S ribosomes anywhere in a eukaryotic cell?
In the mitochondria?
Wonderful. That is the evidence that the mitochondria is a prokaryotic origin, and it has the 70S. It has its own genetic material. It has its own ribosomes, which are of the bacterial origin. So we know that the energy production in a eukaryotic cell is in the mitochondria, and the mitochondria is of bacterial origin. And we know that hypothesis that says that they came to live together in the endosymbiotic theory, that these prokaryotic cells gave up their independence and they came to live within the eukaryotic cell. And now they are the ones that produce the energy for the eukaryotic systems. Anyway, the bacterial and the archaeal ribosomal RNA, you will find different ribosomal RNAs. So there is a 16S ribosomal RNA, which is found in the small subunit. So there are two subunits. You can see the small subunit of a ribosome and the large subunit. They are represented in the figures here. So there is an RNA, ribosomal RNA attached with the small unit. This is the 16S. And do you know the significance of this 16S subunit RNA? Where has it been used and what has it been used for? The sequence of this, the genes for this, have been used to make your domain system. All the work that Carl Woese has done has been sequencing the genes for this RNA, ribosomal RNA, the 16S, which gives you the differentiation, the genetic differentiation, and the phylogenetic tree that we've created is based on the 16S ribosomal RNA genes. Anyway, the other two ribosomal RNAs that are the 23S and the 5S that are found in your bacterial and archaeal ribosomes, they are found in the large subunit. Archaea have an additional ribosomal RNA, which is called 5.8, and this is also found in eukaryotes. So there's a similarity in archaea and eukaryotes to have this 5.8S ribosomal RNA, whereas this 5.8 is missing in your bacterial ribosomal RNAs. Proteins that are found as part of your ribosomes, as such, if you're comparing, archaea proteins on the ribosomes are similar to eukarya, and they are much further apart in bacteria. So we move on to what we call the nucleoid, which is, of course, your area where your nuclear material is somehow located. It is not contained within a membrane, so it is a very diffused area. So irregular shaped region of bacteria and archaea, which we will call nucleoid. We will never call it a nucleus, okay? Usually not membrane bound, but we are finding some exceptions to this rule also, where a single membranous layer may be found around, but it will never be a true nucleus, okay? Here is where in the nucleoid you will find your chromosome, and you know bacteria, most of them have a single chromosome. And not only is the chromosome located here, but there are some associated proteins, just like they have histone proteins in associated with the chromosome in eukaryotic cells. Here we have some associated proteins that kind of condense the nuclear material in a specific area. Usually the chromosome is a closed double-stranded DNA molecule. So it's a circular molecule with just one circle, and it is double-stranded DNA molecule. Now, when I say, and you look at this area that is so large, you would think, oh my God, this is all one double-stranded circle only. And almost, I would say 1/3 of the cell area is covered with this nuclear material. So this must be a huge, huge circle that is all condensed in this area, right? Okay, so here is a very cute little picture of E. coli that has spilled out all its nuclear material, and you've been able to visualize them under an electron microscope. So if this condensed area looks like this, this is what is the outcome of when you release the DNA from the cell, and look at how much material that has come out of this tiny little cell. Okay, so this is a ruptured cell from which the DNA is being, and I love this picture anyway. So you know that there is a lot of supercoiling of the DNA that is occurring in this nucleoid area, and some nucleoid proteins usually are the ones that help in condensing this nuclear material to a limited area. Now you would say, are the nucleoid proteins histone proteins? They are similar to it, but they still differ from the normal histones that you will find in your eukaryotic cells. We come to a component called plasmids. This is, of course, the extra chromosomal DNA. The cell does not need it, but it does give it some special advantages. And these plasmids are not only found in bacteria and archaea, but they are also found in some fungi. So when I talk about fungi, they are eukaryotes. So some eukaryotes, which are represented in fungi, they can also have some plasmids. We have discovered them there. And these are usually, again, small, circular, closed DNA molecules, which will be carrying some genetic information for the cell. The important part about plasmids is that they do not replicate with the chromosome. They are not tied with the chromosomal replication cycle. So they replicate independently of the chromosome, and they exist mostly away from the chromosome. But in some cases, the plasmid may integrate into the chromosome. And we've seen this, and we've tried to differentiate the plasmid that can integrate, and we try to call them episomes. But some authors do not use the word episomes. It was coined for plasmids that integrate into the chromosome, but you may rarely see the word episome being used. But we old geneticists always still use the word episomes. Well, these plasmids contain few genes that are non-essential for the cell, and we discovered these plasmids as the ones that give advantage to the bacterium because they become drug resistance. And drug resistance has been the main plasmid gene that has been studied quite a bit. These plasmids may exist in small copies, copy numbers, or maybe in large copy numbers. There may be many representation of the same plasmid in the cytoplasm of the cell. So we do have, some low copy number plasmids and some high copy number plasmids. This seems to be a genetic characteristic that some will always be in smaller copy numbers, others will be in larger copy numbers. And the important thing about these plasmids are, since they are in the cytoplasm, they still get inherited quite stably during cell division. Remember, we have binary fission in the cell where the nuclear division, the nucleus, once it has divided into two chromosomes, one will be inherited by one cell and the other. But these smaller plasmids do not get equally divided, but they get inherited at least stably, that at least one representation of the plasmid will come in one of the sections or the other. Okay. There is a process that we used to call curing, which is, of course, a spontaneous loss of a plasmid. And this would occur if during the division there is no representation of a plasmid, and all the copies went to one of the daughter cells and didn't come into the other daughter cell. The one that did not get a plasmid would be cured of a plasmid. So curing is the phenomenon we call when a spontaneous loss of a plasmid occurs. And we have a lot of plasmids that we have studied, and we have tried to classify these plasmids, and their classification is usually based on their mode of existence or how they are spread and what are the functions that they carry. So I will give you a list of some plasmids that have been studied very well. And I'm one of those who studied some plasmids before. So this is, of course, of very much interest to me. There are plasmids that are called conjugative plasmids, and they all represent what we call the F factor. F stands for fertility. And they are one of the larger sizes of the plasmids, about 95 to 100 kilobase pair size. And they usually are in small numbers. Their copy numbers are usually 1 to 3 per chromosome, okay? And you can find them, these conjugated plasmids, in E. coli or Salmonella or Citrobacter. And their phenotypic feature, that means what will they give the cell the ability? They will give them ability to produce a sex pilus, and they will be able to perform conjugation with some similar species of bacteria. Okay? Conjugated plasmids. Then there are plasmids that I studied a lot when I was a graduate student in the 1970s. That was the hot topic at that time. R plasmids. R plasmids are, of course, resistance plasmids, and these were discovered to be the ones that carry a lot of drug resistances in them. And a very well-known one is the RP4. RP4 approximate size is about 54 kilobase pairs, and it's again in a small copy number, 1 to 3, and these are mainly found in Pseudomonas and some gram-negative bacteria like your E. coli also, and so on. And of course, there is a new one that is found in your gram-positive bacterium called Staphylococcus aureus, and which is the PSH6. This is the one that we don't want it to be spread to others, and these will give resistance to certain. So the ones that is in the gram-negative ones, the RP4, again, is for sex pilus production, for conjugation, and resistance to multiple drugs such as ampicillin, kanamycin, neomycin, tetracycline. And to tell you that when I did my graduate study and collecting specimens from clinical samples from hospitals to collect all gram-negative bacteria and to find if they were resistant to antibiotics, this is in the 1970s I'm talking about, I found 30% of the clinical isolates already having multiple drug resistances. So this is, we introduced the antibiotics to the human population in the 60s, and as soon as in the 70s, these bacteria are fighting back with multiple drug resistance in clinical strains. So that was an alarming discovery, or knowing that this kind of resistance can easily be transmitted in your gut, in your natural surrounding, to other bacteria which are related to them, and conjugation would be occurring there. Then a very important one, which I also think is the ones that are the col-E plasmids, the col plasmids, colicin plasmids. This is a colicin, which is of course antimicrobial compound that this is like, combating within in nature. When one bacterium is growing in some area, it wants to kill the others because there's limited supply of everything. So they start to produce what we call colicins. And E. coli are the only one, which is the col is from E. coli. So these are usually the colicin plasmids are in E. coli. And these are very small plasmids, where about 9 to 10 kilobase pairs, and they are in large copy numbers because they are small. They can exist in large copy numbers in the cytoplasm, and they produce colicins, which are missiles, or ones that will kill other bacteria around them. Then we have virulence plasmids, which are also very interesting, and you may have heard of the TI plasmid if you've studied with your genetics class. TI plasmids are studied there. It's found in Agrobacterium tumefaciens, and this is, of course, produces tumor in plants, and this is, of course, a virulence plasmid. We have an enterotoxin producing plasmid also. We call it ENT, which is also a large plasmid found in E. coli. Then these are most fascinating to me is the metabolic plasmids, and these are usually found in Pseudomonas species, CAM and the tall plasmids. These are huge, humongous plasmids. These are the very good ones, where this plasmid, CAM plasmid, is needed for camphor degradation. And this one we have discovered as a toluene degradation. Where do you think these plasmids would come in handy in our environment?
In oil spills?
In oil spills, where you want to kill out oil spills, or even in your soil, where these oils are released. These bacteria in nature are definitely the clearing agents that will clear out these camphor, toluene, or some kind of compounds that are very hard to digest away by other bacteria. So your Pseudomonas being found in your soil, and now is being used as an agent to clear up oils or other compounds that are not easily digested by others. We talk finally of bacterial endospores. And this is a property that is endowed to a certain types of bacteria only. So if you ever hear the name of Bacillus genus, you know they are all endospore forming bacteria. Another group that is also endospore forming is which one? Clostridium. So Bacillus and Clostridium are the main groups that are spore forming. Both of them are considered to be the ones um, that are rod shaped, but you would say are endospores only formed in rod shaped bacteria? No, there is a Sporosarcinia, which is a cocci, and that can also produce endospores. So why do I say that this is a very good property that is found only in very few bacteria? Why is it supposed to be such a good property? What is so special about forming a spore?
They're highly resistant.
They're highly resistant to what?
Environmental changes for survival.
Such as
Heat.
heat, UV, chemicals, a lot of environmental stuff that would kill most bacteria. So these bacteria, if they are capable of resisting all these environmental conditions, what is it that we are looking at? They are the ones that have the highest survival on this earth. Okay, so they can survive for ages without having any metabolic processes going on in their spore. They can be like inert, dormant structures. They can come back to life form and divide. But when they are in the spore form, a free spore, they have taken, material from these long-term digs where they are looking at, eras of Earth in terms of millions of years. They've found spores from there, and if they can find the right conditions for their growth, and when they've been brought back into the lab and given the conditions, they can germinate and start to make cultures. So we are looking at these spores as dormant ones, which may be a best survival as the earth has gone through lots of changes in environmental conditions. These spores may be the way to survive for these bacteria to be around us. Okay, so actually you will find the spore being, since it is an endospore, it has to be produced inside the cell. So it starts as inside the cell, and the positions of this where the spore is produced is also a genetic property. So some bacteria will produce a central spore, some will produce subterminal, some will produce a terminal spore, and some will have a spore that the diameter of the spore is greater than the cell diameter. So there is a swelling, and we call it a swollen sporangium, where it will give you a special structure. So having special positions or the way the spore is sometimes gives us cue in the identification of the bacterium also. Once these spores are produced, the vegetative cells disintegrates and only the spore survives, which will then be called a free spore. And this free spore will be very resistant to heat, radiation, chemicals, and even drying. So here is a picture of what the spore is composed of. So this is various layers that surrounds your spore. So the spore is surrounded by a thin covering, which we call exosporium. So the outside is an exosporium. And then inside it is a thick layer of protein that forms the spore coat. So then there is the coat, the outer coat as we call it. And then there is the cortex, which is under the coat. And that cortex is made of the peptidoglycan. So there is peptidoglycan in the cortex of the spore. Well, another thing that I have to mention here, the known genera that we know that are capable of making endospores, they are all gram-positive bacteria. So it seems like nature is favoring gram-positive bacteria, giving them the ability to survive much more than your gram-negative bacteria, which are quite vulnerable. And they have now found their ways to live within the human being, the gram-negative bacteria, which gives them protection from the other environmental conditions of having no food or no moisture or anything. So definitely gram-positive spore formers will be found more in soil in harsh environments, whilst your gram-negative bacteria, most of them will be associated with the human body, where they can very nicely, cozily survive much better there. The core of the spore will have the nucleoid. That means the nuclear material is still there. Of course, it is needed for it to regenerate into the vegetative form. And also with it in the core will be ribosomes. If there were no ribosomes in the core, protein synthesis would not begin and you would not be able to revive the spore again. So what makes the spore so resistant? There is several hypotheses for this as to what makes the spore so resistant. One of them is that we have found that these spores have a high content of calcium in them, and this calcium usually complexes with a compound that is only found in the spores, which is called dipicolinic acid. So dipicolinic acid, a special compound only found in spores, and these are not, this compound is not found in your vegetative cells, okay? So we know that the dipicolinic acid cross-links with the calcium, and this compound together, a complex of calcium and dipicolinic acid, makes the spore resistant to environmental conditions. Also, small acid-soluble DNA binding proteins are found in the core of your spores, which will protect the DNA from being destroyed also. The core is very much dehydrated also. That means metabolic activity is stopped because of the dehydration there. The spore coat and exosporium also give it special protection. The spore coat is quite thick, so nothing penetrates through it. Transport is stopped. So these are some of the ideas that we would think that makes a spore more resistant to environmental condition. Now, when you would say that when is the spore produced? The trigger for spore production is lack of nutrition. When there is a lack of nutrition, then only a spore is produced. If your cells that are capable of making a spore, you expose them to heat, they will not turn into spore then. They have to be already formed spores, then only they can resist heat. So we know that the trigger for spore formation is not heat, it is not UV, it is not chemicals, it is only lack of nutrition that triggers a spore formation. And the process of producing an endospore is quite slow. It takes various different stages that it goes through, and eventually the free spore is released, which is now a very resistant body. So we start with how the spore is produced. You know that cells are dividing and now they are getting the cue that there is not enough nutrition around them. So their cell division starts with unequal division. So this is when binary fission is stopped, and now you're getting an unequal division where the extension or the cell wall is being laid not in the center, but on one area where a small area will be partitioned away with the developing spore that will have the DNA, a copy of the DNA and some ribosomes already trapped in that area. And then it starts putting layers on it where the septum grows around the protoplast, engulfment occurs so that you have another layer that comes around it and it develops an outer coat. Then a lot of dehydration occurs and you go to the stage 4 of your spore formation, and the exosporium is now, and of course your primordial cortex is now almost complete. And you know the cortex is the peptidoglycan, so the peptidoglycan is still being laid around as a thick cortex there. And then the layer called the exosporium is created. And in the stage 4, calcium is incorporated, further dehydration, single-stranded, the acid-soluble proteins are produced at that point, and the coat layers are formed. Eventually, the cortical layers are laid in stage 6, and finally, the rest of the cell, which is the vegetative cell, disintegrates, and only a free spore is released. Now, this is the most resistant form. At this point, it can survive for ages now. I've given it to you in a very short quick of this, we will talk about what is important in the survival of a spore and what are some of the factors. So let's quickly recap this and say, how does the spore now germinate? So sporulation versus germination. Sporulation is the process where the endospore is formed, which I just showed you in the pictures. Germination is the process where from the spore, your vegetative cell has to come out, which will now be capable of now dividing. So as such, sporulation process normally occurs in hours. Up to 10 hours it will take before the spore will be completely formed. So when the cells get the signal that there is not enough nutrition, they start to go into the sporulation stage, okay? Lack of nutrition or nutrient is the only trigger. And the multi-stage process we just saw. Germination is a process where what does the signal for germination is availability of moisture as well as nutrients. So when you give nutrient and moisture to your spores, they will start to germinate. I won't go into how it is done, but definitely spore will swell, rupture, and absorb the moisture and the nutrients, and the loss of resistance occurs, and of course metabolic activity increases, and this outgrowth of the vegetative cell will occur here. So here are the differences that you can read in terms of what is found in a vegetative cell versus your endospore. And I'm going to stop here. I've already run out of time. So we've completed